Welcome back to the class, children. In today's class, we are going to discuss about the concept of codominance. In the last episode, we have discussed about the intragenic gene interaction. That is, the interaction between the alleles of the same gene, right? So that type of interaction where the alleles of the same gene interact together and affects the phenotype. So that is what we call it as a intragenic gene interaction. So under the intragenic gene interaction, we have discussed about incomplete dominance in the previous class. And as a continuation, in today's class, we are going to discuss about codominance. What is codominance? Before entering into this concept, first we have to know what is meant by dominance. This we have already discussed in the monohybrid cross. So when two homozygous pure breeding varieties were crossed, and whatever the trait which is expressed in the F1 hybrid is what we call it as a dominant trait, and the respective allele is said to be a dominant allele or the dominant gene, and the trait which is not expressed in the F1 hybrid is said to be a recessive trait, and this is the concept of dominance. So when two homozygous varieties were crossed and they brought together in a heterozygous condition, the character which is expressed is what we call it as a dominant, and the character which is not expressed is said to be a recessive, and this is what we call it as a law of dominance. But here it is said to be codominance. Let us see what is codominance. Codominance is a type of inheritance in which both the alleles of a same gene expresses their phenotype in the heterozygous condition. Here, when two homozygous pure breeding varieties were brought together, the F1 hybrid will have the character of both the alleles, both the homozygous alleles, right? Both the alleles of both parents, the alleles of both the parents are expressed equally. The phenotypes of the both parents, right, will be expressed equally without the blending nature, right? So the characters won't blend, but individually they will express the phenotype. So if you see the image, it can be clearly understood. So see, this is a red flowering plant of Camellia species. So it is one of the local plant. So Camellia species, and this is a plant, and this variety gives rise to red colored flower. Whereas there is another variety of Camellia which gives rise to white colored flowers. So this is a pure breeding red variety or pure breeding red Camellia species, and this is a pure breeding white colored Camellia species, right? When these two varieties or when these two species were crossed, so look at this hybrid. This hybrid has the character, the color character of both the parents, right? So here in the F1 hybrid, we can able to see both the colors, right? See here, both the phenotypes are expressed individually. It is not a blending character. The triad, which is not a blending of both the characters, as in case of incomplete dominance. In incomplete dominance, while crossing the red flower with the white flower, the F1 hybrid, what we got is a pink colored flower. But here it is codominance, where the red and the white colored flower, when crossed, they gave rise to a flower which shows both the characters, the red as well as the white. So that is what we call it as a codominance. Here both are dominant, right? That is why both are expressed in the F1, right? So both of them are not recessive, right? Both are equal to each other, right? So red is on par with white, or white is on par with red, so vice versa, right? So that is what here we call it as codominance. So in codominance, the F1 hybrid will show the phenotype of both the parents equally, right? The characters won't blend, and moreover the alleles never blend, right? So we know alleles or the gametes never going to blend, right? So only the traits will be mixed. Here, the triads are also not mixed as well as the alleles are also not mixed. So that is why both the characters of the plants were exhibited, right? So this F1 hybrid shows the color of red colored flower as well as the white colored flower, right? We can able to notice the red color as well as the white colored petals in the F1 hybrid flower. When this F1 hybrid flower is selfed, once again we can able to get the hybrid varieties and the white colored flower in the ratio of 1 is to 2 is to 1, and this ratio once again proves only the characters were mixed. See the word, the characters are just mixed, not blended, right? So when the characters are blended, it comes under the concept of incomplete dominance. Here the characters are just mixed, right? Both the characters are expressed in the F1 equally, right, on par with each other, right? So the ratio comes as 1 is to 2 is to 1, right? Because of the law of segregation, because of the law of purity of gametes, the red and the white colored flowers came into the F2 generation, right? Apart from that, we also got the F1 hybrid varieties in the ratio of 1 is to 2 is to 1. So in this case, both the phenotypic ratio as well as the genotypic ratio will be the same, and the ratio is 1 is to 2 is to 1. And this is a very simplest example for codominance. So in codominance, the two homozygous varieties, pure breeding homozygous varieties, when they are allowed to come in a heterozygous condition, the characters of both the alleles will be expressed equally without any sort of blending, right? So both the characters will be equally expressed. So that is why we got the flower as a mixed flower, having both white petal as well as the red petal, right? The characters are not blended. If it is a blended character, as in case of incomplete dominance, the flower will be of rose colored, right? But here the flower is not rose colored. It is a mixture of red as well as the white petals, right? So that is what we call it as a codominance. So and here it is a comparison between the codominance as well as incomplete dominance. So this, from this we can moreover clearly understand, we can further more clearly understand. See, this is the red colored flower and this is the white colored flower. So when they both were crossed, in case of incomplete dominance, the product will be a pink colored flower, which is a blending of the characters, right? This is also a hybrid, but it is not same as that of parent. It is something different from that of parent, and it is moreover the phenotype does not resemble any of the parent, right? So the characters are totally blended. Both of them are not dominant. So that is why we call it as an incomplete dominance. Whereas if you see the codominant concept, both the characters are equally expressed. We can able to see the red color petals as well as the white colored petals. So in this case, the characters are not blended, right? They are present as such, and the triads are equally expressed, and the alleles for the triads are equally expressed. That is why we call them as a codominant allele, right? They are on par with each other, right? So this image clearly shows the difference between the codominance and incomplete dominance. And in our book, it has been given one more example regarding the codominance, which is in the seed proteins of Gossypium species. Gossypium is nothing but the cotton plant. So in Gossypium species, two different species were crossed, right? Gossypium hirsutum and Gossypium sturtianum. So these were the two species, and when these two species were crossed, a F1 hybrid variety has been developed. So when the proteins, right? So proteins are the macromolecules like DNA and RNA. When the proteins of the Gossypium hirsutum was isolated and when it is subjected to electrophoresis, so it shows a band pattern like this, right? So we know electrophoresis is a technique which is used in the genetics to isolate the macromolecular substances like DNA, protein, or even RNA in the form of bands, right? By applying the electric field, we can able to get the bands. So this image I have given you as a illustration, as an example to understand. So while when the seed proteins of Gossypium hirsutum were subjected to electrophoresis, let us imagine this type of band pattern is obtained. Whereas in the same case, when the seed proteins of the another species, Gossypium sturtianum, the band pattern is obtained like this, right? So these were the two different types of band patterns of Gossypium hirsutum and Gossypium sturtianum respectively. When these two were crossed and F1 hybrid were produced, the band pattern of the F1 hybrid species shows the combination of both the band patterns of Gossypium hirsutum and Gossypium sturtianum, right? So here we can able to notice, so see the number of bands. The number of bands when compared to both the parental varieties, here we can able to see more number of bands. And this proves neither Gossypium hirsutum nor Gossypium sturtianum is recessive. Both are dominant. That is why the hybrid contains the band pattern of both the parental varieties, and this shows the increased number of band patterns, right? So here we can able to see in Gossypium hirsutum we have 7 bands, whereas in Gossypium sturtianum we have 5 bands, right? So when these were combined and the hybrid was produced, the hybrid shows a band pattern having more than 8. So it is about 9, right? So 9 different band patterns were obtained, and this shows both the Gossypium hirsutum and the Gossypium sturtianum alleles for the seed proteins are codominant to each other, right? So this is another example which has been given in our book, right? So codominance means the alleles are equally expressed, right? So neither of them is recessive. Both the homozygous alleles which are specific for a particular trait will be expressed equally, and the respective phenotypes will be expressed equally. That is what we call it as codominance, right? So today we have discussed about the concept of codominance and the comparison between the codominance and the incomplete dominance. Hope this session will be a useful one. Let us meet you in the next episode. Thank you.